So as James said, I will continue talking about sonic hedgehog, but I would like to focus on a different structure, the eye or the visual system in general. And actually, I would like to start maybe with the second or concept that Alain mentioned, is how you sort of make morphogen not moving rather than moving, and then I'll talk about maybe a little bit about moving. And for doing so, I would like to introduce two proteins that I have not, so now wants to work. So, and I would like to focus on two proteins, Chidon and Bach, that have not been mentioned so far, which are a cell adhesion molecule of actually very similar to the roboreceptors, composed of a transmembrane domain, an intracellular domain, three fibronectin kind of domain, and immunoglobulins domain, which have been shown by several labs that can interact with patch, which receive sonic hedgehog. And this interaction between patch and Bocken or Chidon favors and enhance sonic hedgehog signaling in different contexts. Now, the other focus of my talk, as I said, is the visual system. And hedgehog is known to be important for the initial patterning of the eye in vertebrate, and it has been shown that midline-derived hedgehog is important for proximal distal patterning of the optic vesicle, so that in low hedgehog signaling, the two eyes tend to fuse because the optic stalk, the region over here that will give rise to the optic nerve, is not properly patterned, and the vesicles, the optic vesicles itself expands, tending to fuse and give a single eye. In contrast, when high level of sonic hedgehog are expressed at the midline, the optic stalk expands at the expenses of the optic vesicle domain. And this is because sonic hedgehog at the midline control the expression of PAX2, and this in turn has a inhibitory relationship with PAX6, which is expressed in the optic vesicle itself in a very much, in a way very much similar to what actually James has just described in the neural tube. Now, we in the lab were interested in understanding Chidon function, and actually observed that in zebrafish, Chidon is expressed throughout the optic vesicles in an overlapping pattern with PAX6, whereas patch, the sonic receptor is expressed in the optic stalk over here, very much overlapping with PAX2 expression. Therefore, we sort of postulated that this expression pattern, which is really complementary, suggested that the two proteins are not indeed interacting, but they are likely acting independently. And therefore, the model that I showed you before of patch interaction with Bach for enhancing signaling was likely, it was quite unlikely. To start to address this question, what Marco Cardoso in the lab did was designing a morpholino that could interfere with kidon function and overexpress kidon in the zebrafish. And as you can see here, using PAX2 as a readout of what happens to the optic vesicles, we obtain results that are quite the opposite of what I illustrated before. In absence of kidon or with a downregulation of kidon expression, what we observed was an expansion of the optic stalk, whereas with the overexpression of kidon, we obtain a cyclopic embryo with basically two fused eyes. Suggesting that indeed a low level of kidon, what it does is somehow lowering the activity of sonic hedgehog. And indeed, supporting that this is the case, if you incubate embryos that have been treated with a kidon morpholino with cyclopamine that blocks Smoothen function, we basically observed a very a rescue of the effect that the kidon morpholino does, expanding the optic stalk at the expenses of the optic vesicles. Therefore, we concluded that indeed kidon really counterbalance sonic hedgehog activity. And to further prove that this effect is really independent from interaction with patch, what we did is, Marco did, was using the idea of morpholinos which can induce exon skipping, and therefore deleting either the patch binding domain or the sonic binding domain from kidon. So with a morpholino that is designed on the ATG of kidon, he repeated this experiment, observing an expansion of the optic stalk. However, when he deleted the sonic binding domain, he observed a similar phenotype in which the optic stalk is expanded. But this doesn't happen when he deleted the patch binding domain. And indeed, in this case, the phenotype of the morphant embryos are very similar to that of control, suggesting that indeed the sonic binding to kidon is important for balancing proximodistal patterning of the optic vesicles, whereas the patch binding domain is not relevant. Now, there are two ways of interpreting this result. We know that kidon is actually expressed very early on at the midline in the gastrulating embryo, overlapping with sonic hedgehog expression. So one could postulate that kidon might interfere with sonic release from the midline, and therefore suggesting that in absence of kidon, what it could happen is that sonic hedgehog would expand more, and therefore promoting PAX2 expression. On the other hand, one could think that expression of kidon in the optic vesicle itself could limit sonic hedgehog movement across the optic vesicle, and therefore abolishing kidon expression just in the optic vesicle would lead to an increase of sonic hedgehog expression, and therefore affecting the proximodistal patterning of the vesicle. Now, if this latter possibility is true, the restricted overexpression of Kedon just in the eye field would be sufficient to rescue the morphant phenotype. And that is actually what Marco did. He injected embryos with the morpholino in a transgenic line that only express under the control of express GAL4 under the control of the REX3 gene, which is expressed only in the eye field, and then use the WAS construct to overexpress Kedon in the eye field, or to overexpress Kedon with mutated version that lacked either the patch binding site or the sonic binding site. And as you can see in this graph, the Kedon overexpression rescued the effect of the Kedon morpholino, and that is also observed when you overexpress the version of Kedon that lack the patch binding site, but it doesn't occur so when the sonic binding site is missing. And this is further supported by experiments in the chick embryos, in which Kedon has a conserved expression in the optic vesicles. If we use morpholinos that interfere with Kedon expression and downregulate the level of Kedon by electroporating the morpholino right into the optic vesicle only, what we observe is an expansion of the optic stalk at the expenses of the optic vesicle. So this brings us to a model in which we believe that Kedon is expressed in the neural retina, or to limit the effect of a sonic hedgehog that comes from the midline to the region of the optic stalk. In absence of Kedon, sonic hedgehog can be transported along the future neural retinal cells, and this induce a conversion of these cells into stalk cells with restriction of the neural retina, and this lead actually to ocular defect, mostly to coloboma, as I will show you later. Now, to really prove that that is the case, that Kedon can restrain hedgehog expansion, Marcos with Luisa in the lab did further experiments using the chick embryos. And what they did is taking the optic recess region of the chick, where sonic hedgehog is expressed with a very symmetric pattern, as you can see here with antibodies against sonic hedgehog, and electroporate in the region close to the sonic source, either just cherry protein or Kedon protein, and then analyzed the dispersion of sonic hedgehog in the region. And as you can see, the presence of Kedon protein somehow distorts the sonic hedgehog distribution, and sonic hedgehog protein accumulates on Kedon expressing cells, but this distortion doesn't occur when cherry is electroporated. Now, if we quantify this distribution in the most proximal or distal region in which Kedon cells, in which Kedon is expressed, you can see that there is a strong accumulation of sonic hedgehog immunoreactivity in the most proximal cells, but this decreases fairly much in cells that are more distal. And similar results are obtained when, instead of Kedon, we electroporate the mutated version that lack the patch binding domain, but it doesn't occur so with the version that lacks the sonic binding domain, suggesting that Kedon indeed accumulate sonic hedgehog protein, acting like a kind of a sink. Another interesting aspect is that Kedon expression is mostly accumulated at the basal site of the neuroepithelial cells, and somehow this overexpression induces the formation of filopodia-like processes that accumulate sonic hedgehog expression, and induces also an expansion of this basal process that has been quantified here, just basing on the width of the basal site. So based on this, we sort of would like to propose that what Kedon can do on the basal side, expressed on the probably mostly on the basal side of the cells, is controlling the dispersion and therefore the signaling activation of sonic hedgehog, but sort of sorting between apical and basal the dispersion of the ligand in the basal side. This will have no activity, as most of sonic is received on the apical surface in vertebrates through the cilia that are present in the neuroepithelial cells. So now shifting a little bit, our next question was, what is the consequence, or how is Kedon mediating, or how is involved with other molecules that are known to contribute to patterning of the optic vesicle? Work from Michel Brandt lab and from Maria Hernandez in the lab under the guidance of Florencia Cavo-de-Assi when she was still in the lab, have shown that nasotemporal, or if you want, dorsoventral patterning of the optic vesicles in zebrafish is very much controlled by the interaction or the interplay between sonic hedgehog in the ventral side and FGF on the most dorsal side, so that these two morphogens regulate in turn the expression of two transcription factors, FOXD1 in the ventral side and FOXG1 in the dorsal side, which in turn regulate each other. And this interplay of different molecules regulate this patterning of the vesicles, so that and this patterning is really important for future targeting of the retinal ganglion cells axon to their target, the optic tectum, so that if you increase sonic hedgehog activity locally, in the neural retina, you induce a shift of nasal projection in more temporal region. So what Marco actually asked is whether FGF was modified in Kidon morphant, and indeed what he observed was an expansion of FGF8 expression, much in line with the idea that there is an interplay between these two signaling components. So what he then asked whether FGF signaling inhibitor were sufficient to rescue the phenotype of Kidon morphant, and indeed by just incubating the embryo with FGF signaling inhibitors, what he observed was a rescue of the proximodistal patterning of the vesicles with a lower expression of PAX2 as compared to the Kidon morphants. This has an impact on nasotemporal patterning, as probably expected. In fact, if we let grow the Kidon morphant, what he observed was a shift of nasotemporal patterning, an expansion of nasal markers like Ephrin 5A or from nasal to temporal, and a restriction of temporal marker like FOXD1 in the temporal domain that here are quantified, indeed suggesting that there is again a relationship between Kidon function in limiting sonic hedgehog expression and therefore also controlling in an indirect way the FGF signaling, which is further supported by the fact that if one knocked down Kidon expression in FGF mutant, the ATHE mutant in zebrafish, there is basically a rescue of the nasotemporal defect that the ATHE mutant have. And this leads us to a model in which Kidon expression in the neural retina somehow limits sonic hedgehog expression, which in turn regulate FGF signaling and the proximodistal pattern, the proximodistal and nasotemporal patterning of the optic vesicle in zebrafish. As I said, the alteration of Kidon expression in the optic vesicles leads to a coloboma phenotype that you can see here, a failure of optics of the fissure closure, and this has leads in turn to defect of axon growth of retinal ganglion cell axon, as shown here with a transgenic line that uses the atonal 5 transcription factor as a reporter for the route that retinal ganglion cell axon take. This defect over here are likely due to the coloboma phenotype, but they just give me a reason to link with the next aspect that I would like to talk about, that is how sonic move instead in the formation of the visual system. And this is actually work that started a long time ago by the observation that Miguel Torres and then work in Steve Wilson's lab in zebrafish, in which they observed that in absence of PAX2, where the optic stalk is compromised, sonic hedgehog expression at the midline is, instead of discontinuous, becomes continuous. And this actually had a strong impact on the visual axon trajectory formation. This is a tracing in a mouse of the outgrowth of retinal ganglion cell axon from the nerve to the chiasm region, where in mouse most of the axon crossed to the contralateral side, but a small percent of fibers instead remain ipsilaterally and grow in the ipsilateral optic tract. In PAX2 mutant, where sonic hedgehog expression is continuous in the midline, this doesn't happen, and most of the axon actually grow on the ipsilateral side. We exploited this observation to provide the first evidence that indeed sonic hedgehog was able to control the growth of retinal ganglion cell axon. And what we proposed was that sonic hedgehog expressed at the border of the optic recess was acting as a negative cue that could funnel without allowing dispersion of retinal ganglion cell axon to the contralateral side of the brain, so that sonic expression at the border form kind of a corridor in which contralateral projecting axon could grow. As I mentioned, in mammals, there are two retinal ganglion cell populations, one that project contralaterally and a small population that project ipsilaterally. This is better visualized probably in this scheme that actually shows that throughout the retina, contralateral projecting neurons are placed everywhere in the retina, but ipsilateral projecting neurons are only clustered in this ventrotemporal quadrant. Notably, contralateral projecting neurons express themselves sonic hedgehog, but this ipsilateral projecting neurons do not. And this is actually better visualized in this immunocytochemistry over here. Now, it's quite well known that sonic hedgehog expression in the retinal ganglion cells are important for moving the propagation of retinal ganglion cells differentiation throughout the retina, so that sonic hedgehog regulates atonal 5 expression and expand the differentiation of these cells. However, in the ventrotemporal region, sonic is not expressed, but these cells express a transcription factor, ZIG2, and the BOC receptor, which is the homologue or a paralog of KIDON with a similar function. So some time ago, we asked whether or not this absence of sonic hedgehog in the ventrotemporal part of the retina and this BOC-specific expression in this region was relevant for the specification of ipsilateral projecting neuron. And indeed, using Bach mutant, we could demonstrate that indeed, in absence of Bach, retina, ipsilateral projecting retinal ganglion cells, here marked by SIC2 expression, are very much reduced, as also seen in here in this double immunocytochemistry with the SIC2, which is a marker for ipsilateral projection, and islet-2, which is a marker for contralateral projecting axons. As you can see, there is a strong reduction of SIC2 positive cells, while there is an increase of contralateral projecting axon within this region, which is also supported by this retrograde tracing of the fiber from the contralateral optic tract. So we got to the conclusion that Bach, in a very much similar way to what we observed with Kidon, that Bach mediated the restraining of sonic hedgehog signaling in the VTC somehow regulates the balance between ipsilateral and contralateral projecting cells. And this, I won't show you the data, has a strong impact on the innervation of this axon to the lateral geniculate nucleus where these cells project. Normally, ventrotemporal ipsilateral projecting cluster in this region of the DLGN, while contralateral from this region cluster in the very tip of the lateral geniculate nucleus. In Bach mutant, this there is a dispersion of ipsilateral and reduction of ipsilateral retinal ganglion cells and an increase of the cells that project contralaterally. Now, this is related to the role that sonic hedgehog has in the specification of retinal ganglion cells. But as I said before, retinal ganglion cells themselves express sonic, and sonic hedgehog is transported along the axons, and the growth cone, as shown in here in tissue culture of contralateral projecting or ipsilateral projecting cells, you can see that cells that produce a sonic hedgehog have plenty of sonic moving along the axon and in the growth cone. And some time ago, we showed that this transport of sonic hedgehog along the axon is relevant to promote the proper growth of the axon along the nerve at the chiasm region, and again help them entering the contralateral side of the brain. Given that if we block a sonic hedgehog reception in contralateral projecting axon, what happens is that growth cone grow in a disorganized way, both along the axon, enter the ipsilateral side of the brain, and disperse along this region of the ventral side. Most recently, work from Fred Jaron lab has proposed that this expression of a sonic hedgehog from contralateral projecting axon has an additional function of repelling ipsilateral projecting axon at the midline, so that he proposes that right at the chiasm region, with an unknown mechanism, sonic hedgehog is released from contralateral projecting axon, and this strong expression of sonic hedgehog is received by Bach expressing ipsilateral axon to force them to enter the optic tract. This is an interesting hypothesis, but we believe that this is probably not all what sonic might do in a visual system, because in zebrafish, where this contralateral ipsilateral sorting of the fibers does not occur, sonic is also strongly expressed in the retinal ganglion cells, in all retinal ganglion cells. And we observed that the protein transported in the inner nuclear layer where dendrite of retinal ganglion cells are. And we observed also with antibodies against sonic transported along the entire tract, optic nerve and tract. So we are very much interested in defining whether sonic hedgehog has also an important role in dendritic organization, and maybe also in targeting of retinal ganglion cell axon to the optic tectum, and we would like, we are hoping to provide an answer to this question. We're using tissue-specific gene targeting of sonic hedgehog and other components using CRISPR-Cas technology. But while the fishes are growing, we still have no answer for this question. And finally, I'd like to acknowledge the people in the lab that were responsible for this work, mostly Marcos Cardoso, who was a PhD student and now is a postdoc in the lab. I briefly mentioned the work of Maria over here. Marcos was helped by Africa Sandonis, a very which is absent from the picture, a wonderful technician. And to this work also participated in the past Françoise, Cristina, Luisa, Curro, and Florencia, that now is in the UK. And I would like also to especially thank our collaborator, Steve and Gaia in the University College of London, Isabel, that will talk after, and Eloisa Herrera in Alicante, and also Emi Okada, who provided us with the Bach mutant. And thanks for the attention.